Hi, thanks so much for inviting me to talk. I'm really happy to be here. So one of the major goals of my career is to try to help improve predictions of future climate change. And in the case of thinking about carbon cycling and how CO2 can enter the atmosphere, we really need to better understand soil processes. And if we can understand soil processes better, we can potentially incorporate them into models and help improve these predictions. So one of the things that I'm really focusing on and trying to improve these, our understanding of soil processes is fungi. And so I have to say, so I've devoted my life's work to fungi. And there are some people out there who just really, really, really love fungi. I think it's a functional trait. And I think there's a gene in there. And I'm not one of them. I like fungi intellectually for what they do. All right. And one of the reasons that I like them is because they drive many ecosystem dynamics, carbon, nitrogen, phosphorus cycling, and more. For this talk, in the interest of brevity, I'm just going to focus on carbon cycling. And I'm going to talk about some of the traits that fungi have that can influence carbon cycling. All right, so one of the things that fungi can do is, they'll decompose dead material like plant litter. And they do this for energy. And as a side product, they'll release CO2. All right, now, of course, that's a greenhouse gas. So this activity is important to consider in models. Now, fungi can also do something else. As they're breaking down material, collecting the carbon from it, they can incorporate it, into their own biomass, especially into cell walls. And that can have a different effect on ecosystems. So some fungi, they'll produce biomass that is itself very difficult to degrade. And so here is an example of, these are rhizomorphs, which are really big hyphae of this fungus Armillaria. And it's actually woody. You can pick it up and crinkle it. It's really tough. And so if the fungi are incorporating carbon in material like this, then that actually might store carbon in there, especially if that material, once the fungi dies, kind of stays in the soil for a while until it's decomposed. And so, of course, if carbon is stored in this biomass, in these residues, then that CO2 is not going up into the atmosphere, and that's something that we need to consider as well. Now, this may seem like a trivial process. We think of microbes as often being very small, how much carbon could be in their biomass. But I think it can be very important. If you look at carbon compounds in the soil, it looks like the majority of carbon compounds in the soil come from microbes, right? They were once part of microbial cell walls and cell membranes. And so that carbon that enters the soil can build up over time, right? And that's important to consider. All right, and then fungi have another function that I'm going to talk about, and that is helping plants grow. All right, so now there are some fungi, and they've been mentioned already, called mycorrhizal fungi, and they can form relationships with plant roots. So they'll colonize them. Here's a picture of some roots colonized by this mycorrhizal fungus. Right, here's all the fluffy stuff is the fungus. And these mycorrhizal fungi, they'll grow out into the soil, they'll mine the soil for nitrogen and phosphorus, transfer a portion of that to the plant, and then that improves the plant growth, because they're basically fertilizing the plant. And as the plants grow more, they're drawing more CO2 down into their biomass, and so that can have a different effect on CO2 going into the atmosphere. Right? So we have three very different potential effects of fungi on just carbon dynamics here. And one of the things that I'm interested in is, what are the traits that can influence each of these processes? And then also, how are they distributed among different fungal species? So if all these traits are distributed somewhat equally, then we maybe don't need to worry about what kind of fungal species are in the ecosystem. But if they're sorted out by species, then we might. Now, we know already that there is some sorting because only a subset of fungi can form these mycorrhizal relationships. But what about these other functions here, like the production of recalcitrant carbon material in cell walls versus decomposition? How are they sorted out? All right, well, we know from looking at just the natural history of fungi that there might be differences in traits among kind of what I'll call morphological groups. All right, so fungi actually are incredibly diverse morphologically. So here are these like filamentous fungi. They're what people usually think of when they think about fungi. They form these like rigid tubular hyphae. Now, in this case for my talk, I'm going to specify free-living filamentous fungi because these fungi are the ones that don't form relationships with plants. They're not parasites. They're not pathogens. Right? They're just classic decomposer fungi. Now, we also have yeasts that are fungi, and they look quite different. So they're actually often unicellular, and they reproduce by budding. And when I started really thinking about traits, I hadn't thought a lot about yeasts, but I've become a big fan. One of the reasons that I like yeast so much is because it seems like they're very, very stress tolerant. They're very tough. And so we'll see them growing on glaciers, in incredibly dry deserts, places that are normally very difficult for other fungi to grow. All right, so I feel like these are stress tolerant just from observing where they're growing, looking at their natural history. All right, and then we have mycorrhizal fungi, which I'll say is my favorite type of fungus. I like how they grow on plants. And they have, specialized structures that colonize plant roots. All right, so 3 different morphologies. All right, so what sort of functional traits, then, are distributed among these groups? All right, well, one of the reasons that I'm interested in this, of course, is because of these feedbacks to CO2 that we might see happening. Now, I'm really interested in climate change. Now, climate change has different effects in different regions, but often it produces more extreme conditions. For example, in the Southwest where I'm working, we might expect drought to be more common. In other areas, we might find more heat waves, more extreme temperatures. Right? So in many areas, climate may become more extreme due to climate change. Well, I mentioned that yeasts in general seem to tolerate extreme conditions. So it's possible in those cases that yeast could respond positively. Now, in areas where the conditions are more moderate, then we might find more free-living filamentous fungi doing well, say. Okay. All right, so one of the questions that I have then is, what are the particular response traits that might direct these differences in distributions that we've been observing? What is it specifically about these fungi that is determining how they respond to climate change? All right, then I'm also very interested in effect traits. So I mentioned that there are a few different ways that fungi can influence carbon cycling. So there might be stronger ability to decompose recalcitrant carbon, right, versus more production of recalcitrant carbon in the fungal cell walls. All right, so these are 2 different functions, 2 different effects. I'm interested in finding out what are the sort of traits that might influence these specifically. What sort of genetic or physiological traits determine whether a fungus decomposes recalcitrant carbon or produces recalcitrant carbon? And then one of the most important questions I have is, how are these response and effect traits linked together, or are they? And this is why I want to know. All right, so if there are linkages between response and effect traits, then we might expect that if there are shifts in fungal communities in response to climate, there could be a corresponding effect, shift in the effects. So let's just say that the response traits are not linked to each other. They're kind of random. So if you have an extreme climate, the yeast might respond positively. If they can decompose recalcitrant carbon and produce recalcitrant carbon, there might not be a net effect on carbon dynamics, right? That's if the traits are just randomly linked. Now, if there's strong linkage, though, between certain response and effect traits, then we might actually see a change in carbon dynamics in response to climate. So let's say that free-living filamentous fungi are linked to a stronger ability to decompose recalcitrant carbon, and that link is strong. Whereas yeasts, say, might produce more recalcitrant carbon, and that link is strong. Then if you have the fungal community being sorted by climate, then you might see a shift toward one or the other of these functions. Okay. All right, so, and then ultimately, that could affect soil carbon storage, right? So let's say you have an extreme climate, it's selecting for yeast to produce more recalcitrant carbon. You might find an increased soil carbon storage, and then that could result in a negative feedback on climate, right? Whereas if you have more moderate conditions and there's a linkage, you could actually have a positive effect on climate because more CO2 will be released. All right, so these are the sorts of questions that I'm interested in, and I wanted to know whether indeed these traits might be linked to one another, right? So we can help predict future climate change. All right, so what are some of the traits that are linked to ability to decompose material? Well, fungi, like a lot of other microbes, if they want to acquire resources from the soil, they'll produce enzymes that they release into the soil, extracellular enzymes, and those enzymes will break down material, and then the fungi will take up the products. All right, so now there's a certain family of extracellular enzymes that fungi can produce, and only certain fungi produce these, actually. This is called lignin peroxidase. So lignin peroxidase, these are enzymes that can go into the environment and break down the compound lignin. All right, so why is that important? Well, lignin is the second most common component of plant biomass, and it physically and chemically protects other parts of the plant biomass. So it'll protect cellulose, right? So if there's a lot of lignin in a dead plant litter, then that will slow down the decomposition of other materials. All right, so one of the thoughts is that fungi, I should mention that lignin is recalcitrant. It's difficult to break down because its structure is randomly arrayed and very complex. Right. So if fungi are able to release enzymes that break down the lignin, as you can see here, then that releases the other compounds in the litter for the fungi to use. So they might break into this cell here and take up the sugars and the cellulose and so forth. So this one particular trait, the ability to produce these extracellular enzymes, lignin peroxidases, can be very important for controlling decomposition rates in the ecosystem. All right. Now there's another trait that fungi can have that is also important, and that's the ability to break down cellulose. So cellulose is the most abundant plant compound in the world. And fungi can use it for energy pretty easily, so they'll break it down to glucose, and then they'll take up the glucose for energy. All right, so when cellulose is in a leaf material, it's, it can be kind of in like a sheet right here. And so basically you'll have different cellulose molecules all lined up and attached to one another, right? And this is called crystalline cellulose, right, this form. This is how it commonly exists in plant material naturally. All right, so one of the first things then in breaking down cellulose to use it for energy is that the fungi have to peel off these cellulose molecules individually. Now, there is a particular enzyme, extracellular enzyme, that fungi produce that can do this. And again, not all fungi produce it. This is called crystalline cellulase, all right? So it catalyzes the breakdown of these linkages here between the cellulose molecules. And then once that cellulose molecule is released, then the fungi may be able to break it down further for energy. Okay, so another important decomposition trait. And we know the gene that encodes for this, and that's very useful. All right, so then the next step in breaking down cellulose is that once the fungus has this cellulose molecule released, then it needs to break the bonds between the glucose units, and it can use an enzyme called cellobiohydrolase to do this. All right, so it'll break down these bonds. Once the glucose is released, they can use it for energy very easily. All right. So different enzymes control different steps in the breakdown of cellulose. And again, we know the gene family that encodes for this enzyme, thank goodness. All right, so those are a few decomposition traits that seem like they might be important for fungi. Now I'm going to talk about traits that confer stress tolerance. How can they tolerate drought or extreme heat, things like that. All right, so one of the things that fungi can do to improve stress tolerance is to construct this carbohydrate. It's called beta-1,3-glucan. Right? So again, this is not produced by all fungi. Right? So beta-1,3-glucan forms cross-linkages in the fungal cell wall. And it forms cross-linkages between chitin here. Here's a cross-section of a cell wall. Here's chitin, and this is mannose proteins right here. So it'll come in and form these linkages. Now, what it does is it basically strengthens the cell wall, right, if it's incorporated. And we know that this helps improve tolerance to stress, especially drought tolerance. Shima et al. 2008 had a really nice paper where they looked at wild-type fungi and the mutants that lacked the gene to make this compound. And the ones that lacked the gene were much less tolerant of drought. Right? Could only tolerate drought about a fifth as much. All right, so this could be an important trait for stress tolerance. And again, we know the gene that encodes for it, luckily. Now, one of the things that I really like about beta-1,3-glucan is that this material right here, I told you it forms these cross-linkages. It's pretty tough. It looks like it could be a recalcitrant carbon compound. It could be one of these things that when the fungus dies and the material enters in the soil, it might be difficult to break down and could stay and contribute to carbon storage. So why would I say that? Well, when people have extracted beta-1,3-glucan, it's very, very difficult to break down in the lab. It's not acid soluble, it's not alkali soluble. It looks like it's very, very tough and complex because it forms these cross linkages. All right, so it could potentially contribute to soil organic matter formation. So I like this because it's basically a dual trait, like Elena was talking about earlier. So it can influence how the fungus responds to, say, drought. It helps them tolerate drought, but it can also contribute to the production of recalcitrant carbon compounds. Right. So if a fungus has this, we have an idea of what sort of feedback it could cause. All right, so what's another stress-tolerant trait? Well, trehalose is, I like it. Trehalose is a compatible osmolyte. And fungi can produce this. They can replace water molecules in their cell membrane with trehalose. And it helps make the cell membrane more flexible, and that confers freezing tolerance, actually. And it also can help with drought tolerance as well. All right, so if a fungus has this gene for trehalose, we might expect that it could be more stress tolerant. Okay, and then the last trait I'm going to go over is the ability to produce cold-induced RNA helicases, also to tolerate stress. Okay, so under really cold conditions, RNA can form these tertiary structures like this that are stable. Right? Now, this interferes with translation. Right? So what fungi can do is they can produce these RNA helicases that will bind to the RNA and unwind it. And then translation can occur as appropriate. Right? So this is another way that fungi can improve stress tolerance, and again, we know the gene for it. All right, so I'm interested to know then, do different morphological groups have different suites of traits, and how might the traits be linked to one another? Now, the best way to address that question is actually to measure the traits in a fungus, right? Now, we have very few of these data for large lists of fungal species. I mean, it's just hard, it's tough to get. But we do have genomes of fungi, thank goodness, from JGI. So thank you very much, JGI. So JGI is in the middle of a large community project. It's called the 1,000 Fungal Genomes Project. All right, so they're sequencing fungal genomes from across the whole phylogenetic tree of fungi. All right. This is an incredible resource. When I first started doing this analysis, at the time there were 157 whole fungal genomes published. Right? Now it's probably up to 500 or so. Right? They're working really fast. I understand that 157 whole genomes does not seem a lot to people who study bacteria. I think there's probably like 70,000 whole genomes of bacteria, probably more than that. But fungi are really hard, okay? They're hard to work with. So I'm happy with 157. All right, so now I could go into each of these genomes and look for those genes that are associated with those functional traits and see how they're distributed. So what I did was I went into each genome and I counted up the number of gene copies for each of these traits and asked whether they were distributed differently among morphotypes. All right, so first I'm going to talk about these decomposition traits. So this is the gene cellulase GH7. This encodes cellobiohydrase. This is crystalline cellulase, and this is the lignin peroxidase. All right. And then we have gene frequency here, the number of gene copies of each of these traits per 10,000 genes. And free-living filamentous fungi, yeast, mycorrhizal fungi. All right, so what you can see here is that the free-living filamentous fungi in each case really stood out in terms of their genetic composition for breaking down cellulose, crystalline cellulose, and lignin, right? Each of these three things. If you look at the yeasts, all right, they're lightweights in terms, they're not decomposing a lot of tough stuff, at least not the yeasts whose genomes we have at this time. And then the mycorrhizal fungi, also, they're not really standing out a whole lot in terms of their decomposer ability. I was a little bit disappointed in this because I like mycorrhizal fungi and I was hoping they would do it more, but this is just the data that we have right now. Okay, so overall, it seems like if we have free-living filamentous fungi dominating a community, we might expect the decomposition would occur pretty well. Now, what about stress tolerance? Right, well, the pattern looks pretty different there. So here you have the ability to make these strong cell walls, the ability to unwind wound up RNA, and then trehalase, the ability to confer drought and freezing tolerance. Now, in each of these cases, the yeast had a pretty high gene frequency number. Right? So the yeasts in this case are pretty stress tolerant, it seems, from just looking at their genetic components. Now, that is in line with what we've observed from the natural history there in these really extreme areas. The free-living fungi didn't really stand out as being particularly stress tolerant, and neither did the mycorrhizal fungi. Right. So what's really interesting here is that we're starting to see these kind of suites of lifestyles, basically, for these different types of fungi. So we have, on the one hand, strong decomposers that can't tolerate very difficult conditions, and on the other hand, we have these really tough fungi that can't decompose very well. All right. Now, the mycorrhizal fungi, from just looking at this complement of genes, looks like they're not good at either of these things, but I just will say mycorrhizal fungi have a lot more going for them. They have this relationship with the plant, and the plant will give them carbon. I just didn't look at those genes for that. Okay, so it looks like these genes kind of sort out by these morphological groups. Now I want to know kind of how well each of the traits is linked to one another. Right. So I can do that by basically performing a phylogenetic independent contrast, comparing like the frequency of one trait, of genes for one trait versus genes for another trait, see whether they're positively or negatively related to one another. All right, so this is just one example. This right here is the gene frequency for cellobiohydrolase. All right, and this is the gene frequency for crystalline cellulase. And each of these individual symbols here is a genome of a different fungal isolate. All right, so you can see here that if a particular fungal isolate tended to have a lot of genes for cellobiohydrolases, they also tended to have more genes for crystalline cellulases. Right? So these traits were positively linked to one another. Now, when you think about it, this makes sense a lot. Right? So if a fungus is going to be investing in the production of cellulases to peel those cellulose molecules off the matrix, then they might as well also invest in enzymes that will break it down so they can use it for energy. Right? So these are positively related. Now I went through and I did the sort of analysis for every pairwise combination of all the traits I've talked about. I am not going to go through them all with you. Right? I'm going to cut to the chase because it can be pretty boring. All right, so I'm just going to show you a summary of how the traits are linked or not with one another. All right, so here I'm showing relationships between the 3 different decomposer traits that I looked at. Now, each of these nodes represents a different trait. The size indicates how widespread that trait was in the phylogeny of the fungus. And then the width of this line indicates how strong the relationship was. I only show lines if there was a significant relationship between them. We have positive versus negative. So right here, these 3 decomposition traits were all significantly and positively related to one another, right? They tended to co-occur. And also, they tended to reside in the free-living filamentous fungi. I showed you that in the earlier graph, all right? So these are the decomposers, it looks like. All right, what about the stress tolerance traits? Well, they were also positively linked to one another, all right? So if one fungus had beta-1,3-glucan synthase, it was likely to have the other two, all right? So this is a suite of traits that might form a lifestyle. And again, these traits tended to reside more in the yeasts, as I showed you before. All right, so we have these two different functions sorting out. All right, now, what about relationships between stress-tolerant traits versus decomposer traits? Are they linked or not to one another? Well, there was a moderate negative relationship between some of these stress-tolerant traits and some of these decomposer traits, right? It wasn't very strong, the negative relationship, so I don't want to make a whole lot out of it, but people ask me about it, so I put it here. So there's a slight tendency if a fungus had more of these stress tolerance traits that it had fewer of the decomposer traits. All right, so altogether, because it looks like these traits are sorting out between different groups and they tend to be linked to one another, then we can start forming some predictions for how these traits might influence feedbacks on the carbon cycle. All right, so let's summarize what we found out. So you have climate changing. Let's say we have climate becoming more extreme. Well, we might expect, yeah, the yeast would be selected for because they have these more stress tolerance traits. Whereas the filamentous fungi don't have as many of these gene counts, so they might be more selected for under moderate conditions. Now, we know that these filamentous fungi tend to have a good representation of lignocroxidase genes and cellulase genes, so we might expect that under moderate conditions that we might have this function be selected for, right, because these are linked. The yeasts, on the other hand, don't really have these decomposer genes as much. Instead, they're producing, because they're producing this beta-1,3-glucan, and it also may contribute to recalcitrant carbon storage, then this might actually end up contributing to just more production of recalcitrant C in the ecosystem. And then the effects on soil carbon storage might be something like this, where if you have more decomposition of this tough material, that you'll have, you'll have decreased soil carbon storage and increased CO2 to climate, to the atmosphere. And then on this other side, you might have more increased soil carbon storage and then a reduction of CO2 being released to the atmosphere, right? So these are two very different potential feedbacks on climate. And I would not have predicted them necessarily if I didn't know how these traits were distributed among the groups and linked to one another, right? So I'm excited about these kind of predictions, and there are many, many others that we could potentially do. And one of the things that we're working on in my department at UCI is we're starting to put these trait relationships into trait-based models to try to predict ecosystem functioning. And so I'm very excited about that possibility. All right, so now this is kind of what I've been talking about for this one study. Now I want to shift a little bit and tell you a little bit about this NC's working group that I'm in, all right, to give them a plug, because I think it's good for you to know about it. All right, so I'm in this NC's working group, and we're building a database that we decided to call Fun to the Fun, and I think it stands for fungal functional traits, right? I was not around when they came up with this name, but I like it. All right, so the deal is that we have, all these different people here. There are probably more that I've missed. We're coming together. We represent different functional groups of fungi. So we have mycorrhizal people, we have endophyte people, we have decomposer people, and we're just assembling all the traits that we can think of that might be useful. And we're all coming from different backgrounds, so we're interested in different types of traits. And so we put together this database, and it's available online. So on the WordPress website for this group, I posted the link to it, so you can check it out if you want. Here are some of the traits, just broad categories of traits that we've included. All right, so what we did, one of the first days that we met, we all voted on what traits we were interested in, and we made a big list, and then we just went down from the top and just found traits that actually had data. Right, most of them don't have data. But these are the traits that we were interested in that had data. So we're very interested in growth rate of fungi, and in this case, we can look at hyphal growth rate. So we have that. Mycorrhizal fungi, they can have different morphologies and how they move, grow through the soil. And so we have that. We have the size of spores. We have traits of mushrooms, basically, for the fungi. That's one of the easiest things to measure for fungi. We're lucky in that respect. So we have how the, this is one type of mushroom that develops, how that develops, what's the size of the mushroom, how it respires, what's the chemistry, like how much chitin does it have in it. All right, we also have melanin content of fungi. We just lost our heads over melanin content. We love it. It was a very, very top trait that we voted for. And I know that seems pretty random, but melanin actually, we're so interested in it because it has a lot of different functions for fungi and the ecosystem. So melanin is this really weird, very complex compound. It's a pigment, and it can protect fungi against UV damage. It can also protect them somehow against drought and freezing and many other types of stresses. So that's really neat. But also, there have been some really beautiful empirical studies where people have tried to decompose dead fungal material with different melanin contents. And if the fungal material has a lot of melanin in it, it decomposes a lot more slowly. All right, so this is one of these sort of dual traits that links response and effect. All right, so we're really into this. We also have tissue stoichiometry. I think we have stoichiometry people here, so we want to include that. Extra enzymes produce what kind of substrates the fungi use, like cellulose or other things. We have tons of functional gene counts from JGI's genomes. We have trophic mode. This is whether the fungus is a pathogen or mycorrhizal or symbiont or free living. And then we also have functional guild. So these are just kind of our ideas of different functions that plants have, like ectomycorrhizal fungi, or vascular mycorrhizal fungi, endophyte, and so forth. All right, so we've put this all together, and you're welcome to look at the data if you want. We're still building it. I think right now we have a few thousand species in this database, but it's a start, and so we're happy to contribute however we can. All right, and that's all. Thank you very much. Yeah.
So you definitely make some decisions about traits. My questions are about, so how did you pick those 6 traits?
Yeah.
Why did you choose gene frequency as the measure of trait state?
Yeah.
And given that the traits co-vary so strongly with taxa, I mean, can you just use taxa to predict and not need the traits?
Okay. So we'll do this. Okay. So how did I pick these traits? Well, I picked the traits. I actually went back into the cell biology literature from the 1970s. And I looked for gene annotations that were supported by mutations, like experimental tests of mutants versus wild type. If there was good evidence supporting a particular gene having a certain function, then I looked at it. So that was one of the strongest filters. And then I also just kind of used, from my knowledge of ecosystem ecology, what might be important. So like lignin protecting other compounds. So that was, but there are many, many other traits. There are so many others, right? These are just ones I happen to feel best about. The second one was
gene frequency as opposed to something else to measure state.
So the genome size of these fungi varies incredibly. All right, so these are tiny, tiny genomes. And once you get up to these orders, they're like so big. And so when I just use gene count, everything is positively correlated to one another just because the ones that have the larger genomes just tend to have more genes of everything. All right, and so I didn't want to have that sort of specious relationship, so I calculated gene frequency. But you could do it the other way. Now, the other thing that Brendan mentioned was that there's really good phylogenetic relationships for a lot of these traits. And I think that's so exciting because I think that is exactly right. You don't need to know exactly what species we're talking about. We can just have an idea of how the traits are arrayed phylogenetically, and then we could use other units to be modeling them, something like that. Is that what your question was?
Yeah, so I mean,
Yeah.
do you gain anything by knowing the traits? So you started out knowing that yeast and filamentous fungi are different.
Yeah,
Isn't
yeah.
that good enough?
Yeah, I know. Oh, exactly. So there are actually models that have functional groups in them. And so we can, so that's wonderful. This is really informative. So now you can have like 3 different functional groups or so in the model, which is tractable. And then you assign the traits that way. It's not necessarily a trait-based model, it's a functional group model.
I see.
But yeah, you can absolutely do that.
Thanks.
Yeah.
So I have time for one more question from Ken.
Yeah, it's great to be able to see the correlation between the expressions of traits.
Yeah.
I wonder, do we also have information about the expression of traits under different environmental conditions, for instance?
No.
Because that's what we need to be able
I
to be predictive.
Yeah, no, I know. I think you're exactly right. So this is just all genetic potential, right? And it's gene frequency, which so it's something that people use, but it's hard to really come up with a very firm reason for looking at gene frequency, right? So people they have ideas of why gene frequency is important, but that hasn't been thoroughly tested, right? So I don't know whether these genes are being expressed and under what different circumstances. I think that's a very important next step. And I'm glad there's lots of room for improvement in this field and on this study.